a junior here at Wesleyan, and I'm a bio major, and today I'm going to be talking to you about how to properly plate and grow your bacteria. So most of you may think that this is not something you guys really need to know, but I think it's really important that you understand the process so that you can truly begin to appreciate the scientists and the doctors in the society, that they can truly learn how to properly grow bacteria and understand what they do so that they can better treat us for our illnesses. And you may think that bacteria are actually very small and very harmless, but they can actually do quite a bit of harm if they get inside our bodies. So it's important to understand this whole process so that you can better take care of yourself. So when you're growing bacteria, there are 3 major steps that you must follow. It's prepare, plate, and grow, or PPG, so that you can better remember that. So it's preparing yourself and your workstation, choosing the plating method, and then growing your bacteria in the proper growing conditions. So first is preparing yourself and your workstation. When you're in the lab, it's always important that you wash your hands before going in and after being in the lab so that you're not bringing in contaminants or taking them out with you. It's also just as important that you dress appropriately by wearing long pants, closed-toed shoes, a lab coat, and even sometimes safety goggles. And then finally, when you're cleaning your workstation, all you have to do with that is wipe it down with ethanol and let it dry completely before starting your work. The second step is choosing your plating method. Before I start, I just want to explain, in the top right-hand corner, you have this inoculating loop, and that's just a metal loop that you use when you're actually putting your bacteria on the plate. And then when you're choosing your plating method, there are 3 main methods. The quick method, as you can probably guess, is the fastest method of plating your bacteria. And all you do with that is you have your plate on the counter in front of you, and you very quickly swipe on the plate with your inoculating loop covered in bacteria. The second method is known as the lawn method, which is a little bit more time consuming, but will give you very good results. It will give you what's known as a lawn of bacteria so that it's completely covered. And all you do with that one is you very slowly cover the entire plate with your inoculating loop, and then you turn that plate 90 degrees and then do the exact same thing over again so that when you do grow your bacteria, you see that complete coverage. And then the final method of plating your bacteria is known as the isolation method. And this one is the most time consuming, but it gives you the best results to isolate your colonies of bacteria. And how you do this one is you're first going to divide your plate into 4 sections, and then in the first section you're going to do a quick method, and then draw a line with your inoculating loop from the first section to the second section, doing another quick method, and then repeating that from the second to the third, and then from the third to the fourth. And then what you'll see once your bacteria grow up, as you can also see from this image right here, is that that 4th quadrant is going to have the least amount of bacteria so that you can better study them as individual groups. And now step 3 is letting your bacteria grow. And for this one, bacteria are a lot like people in that they need a lot of the same nutrients that we do, such as iron, nitrogen, potassium, magnesium, and other things like that. And basically what you want to do is you want to choose a plate that most likely we use LB nutrient plates, which contain all those nutrients that the bacteria need. And once you've plated your bacteria using one of the 3 methods I mentioned earlier, you're going to place your plate into an incubator at 37 degrees Celsius overnight. And the reason we use 37 degrees is because that's body temperature for humans. And then once you're finished letting your bacteria grow overnight, you can place them in the refrigerator until you're actually ready to study them. Now, since I've talked about the 3 plate methods, which one do you guys think was used here in this image?
It's isolation.
It is isolation, correct. Thank you very much for answering. So in conclusion, you know everything that you need to know about plating and growing your bacteria. The 3 main steps were PPG, so prepare, plate, and grow. Preparing yourself in your workstation, plating your bacteria with the right method, and then letting them grow overnight in an incubator. And I know, again, that you guys might think that this isn't really something you really need to understand or know, but I feel it's very important that you guys understand the whole process so that you can begin to truly appreciate scientists and doctors in society so that they know exactly what they need to do to help us get better when we're sick. Thank you all for coming to my presentation.